Hello and welcome to this video on plague and human evolution. There is a proposition that humanity has stopped evolving. Rather than respond to the environment, we change the environment to our needs or wants. At least, to some extent, this is true. We have vaccines to stop infections from killing people, antibiotics to deal with bacteria, heating and cooling for temperature control, a relatively stable but arguably very expensive food supply system that prevents starvation for the most part. All these would be pressures that humanity has to overcome if we did not have the ability to prevent them. The only way to overcome such pressure is evolution. At the same time, we must recognize that humanity's ability to overcome environmental pressure has not always been the case. An example that we still see the effects of today is plague, a disease, or perhaps better to say a group of diseases, that have altered human genetics, and we still see those ramifications today. That is, we still see the consequences of plague altering human genes going back hundreds, if not thousands of years. And to be clear, when we say plague, we are referring to the Black Plague or Yersinia pestis. There have been a variety of plagues that have occurred throughout human history, some more recent than others. But let's talk specifically about Yersinia pestis, the Black Plague, Black Death, bubonic plague, and other names. Yersinia pestis is a Coccobacillus, that is a particular kind of bacterium. It's shaped in a particular way, hence the name Coccobacillus. It's considered a facultative anaerobic bacterium. It means that it can work either with or without oxygen. Also, when you stain it, it produces a particular weird shape. On top of all of these bizarre features already, Yersinia pestis has another feature that makes it particularly dangerous. It has what is described as an anti-phagocytic slime layer. What it basically does is prevent phagocytosis by cells. In terms of identifying it and making sure you know what you're looking at least at a microbiology level, you're going to test it for things like urease, lactose fermentation, and indole use. Urease, lactose, and indole basically being various different petri plates or agar plates you can make. It also likes to grow at a slightly higher temperature than usual. A lot of bacteria tend to be cultured at somewhere between 20 and 25 degrees. Instead, this is between 28 and 30, and it does rely on a fairly standard pH, a little above neutral. On top of all of this, it's very vulnerable to UV light and temperatures above 40 degrees Celsius, which, given what happens when the human body undergoes fever, is not particularly advantageous for it. Admittedly, it's also not particularly good for human health above 40 degrees Celsius, so take your pick there. It's either damning the host or damning the bacterium. There are 11 different species that all come under the Yersinia genus, and because of this, we need to look at this in somewhat more broader terms, but fortunately, we can narrow that down quite a bit. Yersinia pestis being the most common, but it's one of only 3 that cause disease in humans. In terms of diseases in humans from Yersinia pestis particularly, it is a nasty disease because of its ability to not only avoid but suppress the human immune system, which obviously means it's a very nasty pathogen. Part of what happens here is, that obviously mentioned phagocytosis resistance due to that slime layer, but also the fact that it makes it more difficult for the human body to generate antibodies. And then of course we have how it is that we get infected in the first place. Now historically, the plague has been infamous is perhaps a better word, for its ability to infect humans, but also how it came to be infected. The general understanding is that somewhere in the far eastern steppes of the Mongol horde, small rodent-like creatures were largely responsible for the plague starting. It then spread to fleas from these creatures, and then from fleas to the Mongol hordes, and Mongol hordes brought it across to Europe, and through the rest of Asia, mind you. They weren't exactly a shy bunch when it came to expansion. From this, we then have infecting of humans via something like, say, flea bites, if it's not just exposure in other ways. What happens though is, because you are able to get past the very first layer of the immune system, that is the human skin, you don't activate certain things that should cause the immune system to help fight off viral infection, or in this case bacterial infection. This is plasmin activator. Because of this, and understand it's not a unique phenomenon, it's fairly common for a lot of viruses and pneumonic diseases, you don't get the kind of response you would need to get the body going right away. Following this, because the bacteria is now in the body, and it can survive because it's anaerobic, at least facultatively anaerobic, it also becomes resistant to the first immune cells, the macrophages, that would otherwise have picked it up. The macrophages should phagocytose, or basically swallow up and eat, the Yersinia pestis, take it back to dendritic presenting cells, and then say, here is the target. But no phagocytosis, no dendritic cell function, no ability for the human body to develop an immune response, or at least drastically reduced ability to do so. On top of all of this, even if, and this is the big if here, a monocyte manages to somehow eat one of these Yersinia pestis bacteriums, it's going to start producing certain antigens on its surface, and this makes it harder again for the immune system to deal with it. It takes production of particular antibodies that are largely specific to it in order to be able to do something. Famously, a plague is communicated between animals, including humans, by fleas. As mentioned, and this not just adds to the virulence of plague, but also to its ability to avoid the immune system and stay in circulation. This is one reason why plague was so deadly, and by deadly, we're not talking American school levels of deadly, we mean proper deadly. The Black Death, or plague, is considered to be one of the deadliest pandemics in human history. It killed between 30% and 60% of all people in Europe, the Middle East, Africa, and Asia at different times. Admittedly, those numbers are drastically varying based on different reports, population sizes, accurate record keeping, and so on, but you can basically assume somewhere between a third and nearly 2/3 of all population in an area were gone. Like that. This is a phenomena that put so much pressure on humanity that entire towns, villages, and hamlets were simply gone. Others took centuries to reach pre-plague population levels. Others again never recovered to that level and still remain at a lower population than they were before the plague came through. This is where research becomes somewhat more useful. Now, because so many people died, there is a fairly large amount of human remains, let's say, left to look at and see what happened. Unfortunately, at certain times, this was such a large-scale death that they literally just dug a giant hole and threw all the bodies in there. In other times, they dug several layers to that hole. Essentially, you have different plagues that came through at different times, and they just stacked up bodies, covered them with dirt, and then went, we've got more room, let's throw them in again next time. Okay, slightly glib, but that is actually what happened in very simplified terms. So, researchers have gone and, well, they've been grave robbing. They've taken out bodies from places in Europe, mainland Europe, and in the UK, and they've tried to compare what genes are present and active. There were initially about 350 different genes that were thought to be responsible for some of the immune response to plague. They then tried to figure out, okay, we have these people who died of plague, and this group that didn't, is there any difference? And they found that of these, there's actually a fairly large difference in the genes that have changed between plague and non-plague victims. When they then cross-checked these plague victims from England, or the UK, and victims from Denmark, they found that there were just 4 genes that were particularly important. So they narrowed it down fairly well when you consider what this is. Of all the hundreds of genes they initially found, they narrowed it down to 4, and of those 4, one is particularly important when it comes to the immune system. It is the ERAP2 gene. Now, the name itself is, well, unsurprising. It's endoplasmic reticulum aminopeptidase 2. Calling it ERAP2 or ERAP2 is just easier. What it does is help with immune cells to recognize and then fight off things like a virus. Now, understand that Yersinia pestis is a bacteria, not a virus, but that doesn't mean that the two can't be related. In terms of what they found particularly here, ERAP2 had 2 variants, or 2 different alleles, in the samples they found, and the difference was a single line or single letter in the code that makes up the particular gene. So you have one substitution and suddenly there's a huge difference in what's going on. That difference will either lead to the production of a full-length or a much shortened version of a protein. That difference in ability to make that protein in a shorter or longer version led to a huge change in survivability. In normal conditions, let's say, a shorter version that seems to work just as well as a long version would provide a certain evolutionary advantage. Simply put, you're making the same end result, but you're having to use less material to do so. When plague was present, the full-length version was far more effective, and essentially people who survived were twice as likely to have the full-length version than the shorter version. In terms of trying to actually get real-world numbers to back this up, it's not quite as solid, but they did find that of their 143 samples from the UK, about 40% of the people had one or two copies, depending on whether or not they had a homo or heterozygous copy of the genome, and about 35% of plague victims had the shorter version. This means that those who survived were more likely to carry the bigger version and therefore pass on their gene. Now in Denmark, numbers are similar but a little bit different. So for example, you go from roughly 40% in London before plague to about 50% afterwards, and in Denmark you go from 45% before plague to about 70% afterwards. The next thing here is to talk about the actual role it has to play in terms of the macrophage itself. Remember what we mentioned about how macrophages, they're going to go along, they will phagocytose whatever the particular pathogen is, they'll then take it back and present it to an antigen-presenting cell. When you have these particular alleles or mutations in the genome, you have a difference in the ability to not only detect but then present these antigens, thereby allowing for the human body to be able to more effectively respond to it. While somewhat speculative, it is easy enough to understand it this way. If you have the full size and therefore full complement of functions involved in this bigger version of that particular protein, you're more likely to be able to be more sensitive and reactive to the particular pathogen that's otherwise difficult to find and therefore respond to. If you have two versions of it, well, you now get twice as much if you're presumably not experiencing the silencing of one copy of that gene. However, if you have a silencing phenomena, then just having the full version is going to be more useful. This brings us to the role of evolution and natural selection. As you can imagine, in an environment where a particular pathogen is causing up to half the population to die, well, having something that prevents you from dying, or at least reduces the probabilities, rather important. In this case, the pressure is just that, Black Plague, and the ability to, at least theoretically, clear it out of your body that much quicker. And if you can clear it out of your body quicker, you're less likely to die, and therefore you're more likely to reproduce and pass on your genes. Therefore, well, in short, Black Plague selected for this bigger version or more complete version of that gene, and once you have the more complete version of it, you therefore get more survivability. More survivability, more kids, more of the gene present. And as we mentioned, this is not something that we aren't aware of today. It still affects a lot of people. Now, we will point out that the numbers here are based just on the current population, not population directly derived from the people who would have experienced plague in Britain. So just be aware of this. Previously we mentioned that up to 50% of those that they were looking at would have had two copies of this gene. Today the number's about 45%, and it's been up to 1,000 years since this was a factor. So over 1,000 years, it's basically been diluted down by 5%. That's not a lot of change when you consider how many people have been born, the sheer number of people who have died, the migration issues and everything else. Britain's population is not something that should have just changed by only 5% unless there was some clear advantage to this. But understand, there are some very clear limitations to what we've based all of this on. The most obvious issues are going to be sample size from plague pits and the ability to extract not just viable, but reliable DNA from those remains. Much like in Jurassic Park, where they had to splice in frog DNA of all possible animals, the remains in plague pits will have gaps or holes in the genome that need to be recognized and compensated for. This is not insurmountable, but does need to be recognized. The number of remains from which DNA is extracted is also much harder to compensate for. They do, to some extent, recognize this, particularly by not focusing on just one country. To be very generous here, let's say, by checking against another country, that is, they are able to get a good idea whether or not it's an actual selection pressure, or if what they're observing is just a weird random phenomena. Essentially, it's two isolated independent populations exhibiting the same phenomena. The last thing to note is that humans are complicated, particularly the immune system. It's why we still have no idea as to how to deal with certain autoimmune diseases today. And because of this, there may be a lot more interactions and certainly a lot more complexity when it comes to the cellular response to things like plague that just aren't accounted for by this. But since we're apparently seeing at least the same phenomena across two very different countries that had relatively little to do with each other at the time, there's a good chance that, well, they are the same sort of thing. Even with that, we need to put these findings in the context of the time, and that is, well, in some cases as little as 700 years ago, in other cases more, in other cases less. We need to account for the fact that much of the mortality that was exhibited at the time could be explained by more than just the plague itself. For example, in some countries, for example parts of Northern Europe, had massive issues to do with the weather, and this led to not just crop failures, but, well, all sorts of other problems, and so this might also have been playing into the effect of natural selection. Essentially, if you starve and die, you don't pass on your genes. This does mean that plague may not have been the only thing to affect this. At the same time, you could argue that because these people would be, for all intents and purposes, already made vulnerable by famine, they may have just had an amplified effect from Black Death or plague, meaning that the phenomena we're observing is just an amplified version of what would otherwise have happened regardless. It's a lot of, let's say, questions still to be answered, and as with the catch cry of all research, well, more research is needed, and preferably with funding. We will point out that not all of this is, say, cream and strawberries, or sunshine and daisies, or whichever particular optimistic euphemism you want to use. And it's because the mutations, or perhaps it's better to say, the selection for certain mutations in the genome has not been to our benefit in its entirety. Certain conditions, for example Crohn's disease, seem to be closely associated with the particular ERAP2 gene variant that was selected for through plague. This means that, yes, if we're currently dealing with an outbreak of some sort of pandemic, it's probably great, but if you're just trying to live normally, it's going to create other issues, and there are other autoimmune diseases that are similarly positioned and seem to be related to the ERAP2 gene. So this is both good and bad. We're better protected against a pathogen, but it's a pathogen that's no longer going to affect us. And presuming that you're not an anti-vaxxer, it's arguably a worse trade-off now because, well, we have fairly damn effective means of protecting ourselves without needing to rely on certain genes to be selected for by our ancient ancestors. This does raise questions about what are the diseases that we face that might do something similar to the human genome, especially if and or when, and more likely when, we can respond to them fast enough. Think of how things might have gone without vaccines for COVID-19. If bird flu becomes a pandemic, or fairies become an acceptable part of everyday life. Hopefully we'll find some sort of genetic mutation that explains why it is that fairies exist. But beyond that, well, we're probably not going to do much about bird flu or COVID, or some other respiratory disease of a similar kind. We will just have to rely on either the pharmaceutical industry or natural selection. Thank you for watching this video. If you found it interesting, consider liking, sharing, or subscribing, and please do post any comments, questions, or suggestions you have below.